8:30. That's when that's the time to celebrate vision research. And today we have a very special guest from Iowa, and it is nothing related to the election or anything like that. It will be just a vision research. So I'm really delighted, Bud, that you join us today, and let John introduce you.
Hello, my name is John Hong. I am an MD PhD candidate in the lab of Dr. Krzysztof Palczewski. It is my pleasure to introduce Dr. Bud Tucker, a professor of ophthalmology and visual sciences at University of Iowa, Carver College of Medicine. He also serves as the director of the Ruby Retinal Engineering Laboratory at the Institute for Vision Research, as well as the director and founder of the Stephen Davidesi Translational Vision Research Facility. Dr. Tucker did his PhD training in neuroscience at the Memorial University of Newfoundland in Canada. Then he did his fellowship training in ophthalmology at the Shepard's Eye Research Institute at Mass Eye and Ear Infirmary of Harvard Medical School. His research focuses on a range of topics from gene therapy, cell therapy, retinal disease pathology, retinal engineering, and cell or tissue transplantation. He's dedicated to the mechanistic basis of retinal diseases, as well as the discovery of methods to prevent or restore vision in those suffering from blinding diseases. He has published in over 140 peer-reviewed publications and 6 book chapters. He has published in journals such as PNAS, JCI Insight, Nature Gene Therapy, Human Molecular Genetics, Scientific Reports, Ophthalmology, Nature, Neuroscience, and eLife. He's also dedicated to teaching and mentorship, with having trained many postdoctoral fellows and doctoral trainees, while also teaching in multiple labs and courses on neurology, neuroanatomy, neuropathology, genetics and inherited retinal diseases, stem cell, and tissue engineering. He has received the Kogan Award at ARVO, Ruby Chair of Regenerative Ophthalmology, RPB Catalyst Award, and the NIH New Innovators Award. He has been invited to speak at the Kogan Award Lecture of ARVO, NEI Audiation Goal Initiative of ARVO, University of Toronto, Memorial University of Newfoundland, UCSF, Basel, Switzerland, and Harvard University. With great anticipation, it is our pleasure to have Dr. Tucker present his talk for us today.
Thank you for that very kind introduction, and thanks, Chris and the organizers, for inviting me to give this talk today. As I was mentioning as we had started the banter at the first of the call, we're under a blizzard warning here in Iowa, so the last 4 days we're going to have accumulated on the order of 2 feet of snow. So it would be nice to be in California with you guys in a balmy 40 degrees at the moment. Nevertheless, I'm there in spirit. So what I'm going to tell you about is really my lab's focus over the last 10, 12 years, since the time I was a postdoc or ended my postdoc through to now, really focused on developing treatments for heritable blindness. Sorry, I have no financial disclosures. I don't know why that slide always catches me by surprise. I start off my talk and going to go into the data, and I completely forget that thing is there. Not that this group really needs a lot of introduction to retinal anatomy, but for the sake of completeness, not knowing everyone who's on the call, I thought I would give a very brief overview to start, because it's really important for the diseases we're focused on. So the retina, for all of those who work in the retina are well aware, it is an extension of the central nervous system. Like the cerebral cortex, it's laminated, multiple cell layers, and it's actually in sort of this upside down orientation where light actually has to pass through all of these different layers of neurons before it's detected at the level of the outer segment of the photoreceptor cell, where it's turned into an electrochemical impulse and sent to the brain via the optic nerve, which is made up of the axons from the ganglia cells. Now, what people are probably not as used to looking at, because these are clinical images here on the right, is a normal fundus photo. This is from a normal human. And what you can see here, what I want you to appreciate for the purpose of this talk, is this sort of uniform coloration, this pink-orange coloration with this nice sort of sheen on the inner retina. I also want you to notice the course and caliber of the inner retinal vasculature, which is going to be very important for clinical signs for diseases such as retinitis pigmentosa now in a moment. You can see this retinal vasculature sweeping around the center of the retina, the avascular zone in the middle where the macula is. And the thinner ones are the arterioles, the larger ones are the venules. Beneath the fundus photo here, I'm showing you an OCT, and all I want you to appreciate from this image is that the dark layers are where the cell bodies are. The light layers are really the synaptic connections where all of the axonal extensions and wires are. And finally, beneath that, I'm showing you sort of an odd representation of a Goldmann visual field. I'll show you some two-dimensional ones in a few slides. This is a 3D visual field, and I think this is useful because what it shows is essentially the extent of our visual field, which is the outermost limits of the blob, but the height shows the sensitivity at each of these areas. So as you get closer to the center of the retina where the macula is, you have a much more sensitive or ability to detect lower levels of light and higher acuity. So as patients come into us with inherited retinal diseases, what's happening in this sort of category of disorder is that a genetic mutation is resulting in a loss of those light-sensing photoreceptor cells. And you can see these are progressive disorders. And as the disease progresses and the photoreceptors are lost, the inner retina collapses down on the retinal pigmented epithelium. And as you can see now on the top right, the Goldmann visual field, there's a very constricted visual field at this point. This is a patient with pretty advanced retinitis pigmentosa. If you look at the fundus photo right and under that visual field, you can see two things that are classic signs of RP or retinitis pigmentosa. One is pigmentary changes, known as bone spicule-like pigmentation. These have nothing to do with bone. There are pigment granules up in the retina, as you can see out here. These lacy bone spicule-like pigmentations are classic symptoms or signs of retinitis pigmentosa. The other classic sign of photoreceptor cell loss is narrowing of the inner retinal vasculature. So what happens when you lose all of the photoreceptors, which are normally consuming a massive amount of oxygen from the choroid, the blood supply beneath, the inner retinal vasculature now is the inner retina seeing all this oxygen, so the inner retinal vasculature constricts, and that's a telltale sign of photoreceptor loss. And of course, if you look at the OCT beneath that, you can see outside of this very small hill of photoreceptors right in the center of this patient's retina, all of the photoreceptors are completely gone. Well, genetically, I called this a category of disease because genetically there's actually a lot of different genes that make up this category. And so this is a paper we published in 2017, largely driven by Ed Stone. So these were 1,000 consecutive families seen in Ed's inherited eye disease clinic. So 3,348 individuals seen over 5 years. So it's consecutive. So we really get a representative sampling. Individuals came from 40 states, DC, and 7 countries. There are actually 104 different genes identified in this cohort. 85 of them were photoreceptor specific genes. Now, interestingly, the top 10 genes cause 60% of disease. So the vast majority of disease causing genes are quite rare. The most common we think is ABCA4 is about 400 new cases per year in the US based off of this data set. The rarest being sort of 1-2 new patients a year, genes such as USH1G. Well, how do we treat these diseases if there's 100 different genes? Well, it really depends more on 2 things associated with the genes, associated with the disease, which is the state at which the person's retina is at the time of presentation. And secondly, our knowledge of disease mechanism. So if the person shows up early in your clinic, okay, where they have lots of photoreceptor cells left, we know exactly what the disease gene is and how that gene functions and causes disease, then our ability to intervene early using something like drug or gene therapy is greatly enhanced. Unfortunately, for patients like the individual I just showed you in that montage as it was rolling by, that individual has already lost the vast majority of their photoreceptor cells. So a gene therapeutic approach, which is really designed to prevent disease progression, is not going to be effective. So they're going to need something sort of more invasive, such as a cell replacement-based approach. So for the remainder of the talk, really what I'm going to show you is how our group, use induced pluripotent stem cells, which were first reported in 2006, as you can see here on the left, by Shinya Yamanaka and his postdoc, Takahashi. And we're using these cells, which are virtually identical to embryonic stem cells that we can generate from any of our patients, to evaluate disease pathology, develop gene therapeutics, and autologous cell replacement strategies. And I'm going to do that through a series of case studies, two in particular. So this is two of our patients. These are two young girls. They're twins, fraternal twins, and this was their 6th birthday. So the year before they were 6 years old, they had perfectly normal vision, 20/20. At this visit, they were both 23/20. So twin A is on the left, twin B is on the right. You can see some of those classic signs of photoreceptor cell loss that I had mentioned. So the first thing you can tell here, you don't see any of these bone spicule-like pigmentation, which you wouldn't expect. These patients, there's no form of RP that does this. But you can see a constriction of the interretinal vasculature in both individuals. Now, what's unique in this disease, you can see the macula here is selectively involved. So we get these atrophy right in the center of the child's retina. So this is a unique disease. There's not many diseases do this. These two individuals are still young, so they have still very full visual fields. Now you can see these are the flat visual fields that I have said. So here, the different colors that I'm showing you essentially correspond to different intensity of light. But the thing is important is that they're still pretty wide. These patients still have a lot of photoreceptor cells remaining, despite the fact that they're 23/20. So what is this disease? Well, again, there's very few diseases that do this. First of all, these two children were perfectly normal, had perfectly normal 20/20 vision 1 year prior. At this point, they're both 23/20. There's very few things, again, can do this. Maybe Stargardt disease, ABCA4, in the worst cases, you can have very aggressive disease. But unfortunately, these two kids have Batten disease or CLN3 associated disease, the most common disease-causing mutation being a 1kb homozygous 1kb deletion in the gene CLN3. This is a lysosomal storage disorder. Unfortunately, if left untreated, these children will likely be light perception or NLP by age 12 to 14. Usually within their teenage years, they start to have seizures, and by late 20s, early 30s, this disease is fatal. So this is the worst disease that an ophthalmologist would ever diagnose and take care of. Well, interestingly, with this disease, there's quite a bit of literature, and we know it's a lysosomal storage disorder. So the first thing we did scientifically to try to develop a treatment for this was to develop a model system that we could use to test novel therapeutics. And specifically here, we use induced pluripotent stem cell-derived retinal organoids, which I'll talk about a whole bunch in a few slides. But in this case, what we first did is went and looked for this lysosomal storage defect, which has been reported in the literature. So what happens with patients who have dysfunctional CLN3 is you get accumulation of this toxic autofluorescent lysosomal storage material. And you see lysosomes themselves here stained with LAMP1 or lysosome-associated membrane protein become really bloated and big. So if you look at the top three here, this is in normal individual compared to patient 1 and patient 2, you can see accumulation of this autofluorescent storage material and these very bloated LAMP1 positive lysosomes. So then the question is, can we actually develop a gene therapeutic to prevent disease progression? Right? Because again, the majority of gene therapy is really a preventative, not restorative approach. So in order to do that, the first thing we did was develop a series of vectors. Here's one I'm showing, an AAV vector, just with a constitutive promoter driving CLN3. And the reason we use a constitutive promoter here is this is not cell type specific. This is pan-neural. So all neurons in the retina express this. So having this expressed throughout the retina would actually be a good thing. We used AAV2 because AAV2 in human does hit the different layers of the retina, or the different layers of the retina. We've now switched, and we can talk about that near the end. Other vectors that may be better than AAV2, but sufficient for this talk. You can see here on the right, what I'm showing you is expression in a normal retina. These are actually fibroblasts from an unaffected individual versus fibroblasts from the patient. CLN3 is expressed in almost every cell type you look at. You can see after just 1 week after transduction, we can get robust expression of CLN3 using this vector system. So the question is, well, how does it work in the retina or the brain? So here we made these sort of mini brain retina organoid things on the dish. This was quite a while ago, before 3D organoids were even a thing. So we made these sort of little cerebral organoids in 2D. And what you see here is that normally you would get expression of CLN3 in about 30 days of differentiation. There's very low levels of expression in both of these individuals. After just 7 days of transduction, we get robust expression of CLN3 protein. Well, what about function? Does this gene therapy vector actually function? Now, in order to test this, I'm going back to the model system that I first presented in a single slide, which was this autofluorescent lysosomal storage material accumulation. And what you can see here in 2 of these different individuals, so now these aren't actually the 2 twins. One of these individuals is a compound het for the 1 kb deletion, and the other one is a homozygous for the 1 kb deletion. So the 1 kb deletion patient actually has a much more severe phenotype. But in both cases, what you can see is that transduction of retinal organoids from these patients at 30 days, and then harvest and immunohistochemistry at day 120, you see we can get significant reduction in accumulation of autofluorescent lysosomal storage material. So now we have some evidence that we can make a gene therapy vector to restore the protein, and B, the protein is actually functional and it prevents accumulation of this toxic lysosomal storage material. So the next step is to take this to the clinic, right? Well, how do we do that? Well, we for years struggled with this entire process because the way it would normally done is you would make these vectors in a lab, get this efficacy data, and then you would go to a contract manufacturing organization to produce the virus for you. And then you would take that virus and send it off to a CRO or a contract research organization who, are equipped to do GLP studies to take it all the way through so you could get the safety data you would need to essentially fulfill the requirement of the FDA. Well, the problem with that process is it's very expensive. And so for a single gene like this, or a single gene therapeutic, it would cost us on the order through that process of, $5 million. So this is why treatments that are coming out are millions of dollars per patient, which for a disease that's this rare is below what we like to call the commercial viability threshold. We don't know where this threshold is, but for genes that affect 1 to 2 patients a year, having a commercially viable product is going to be very challenging. So based on that, we decided back in 2014 to 2016, we needed to be able to make these drugs ourselves, and we need to be able to do the testing in-house ourselves to keep the cost way down. So what I'm showing you here is actually the DESI facility that was mentioned in the very first part of my talk during the introduction. This is just a small GMP facility we've built over the last 3 to 5 years. It has two suites in it. The top suite, and I'm showing you here, is an AAV production suite. This is just a very simple, small biosafety cabinet, essentially, that we're walking in. As you can see, they're just all powder-coated walls, low wall returns, with a grated ceiling that's blowing air through HEPA filters. And then within this room, we have just standard cell culture equipment. This hood that I'm showing you here on the end wall actually has a bioreactor in it. So all of our viruses are made in that facility in a bioreactor. So we can, for very low amounts of money, like on the order of $5,000 a batch, produce clinical grade virus that we can then go and take to our animal studies. And what are those animal studies? Well, the animal studies that the FDA requests now for a gene augmentation trial such as this are actually quite simple. It's a 60 animal, 60 rat study. We use rats instead of mice because of the size. And specifically, it's very difficult to get enough blood from a mouse in order to do all of the hematology and clinical chemistry tests that the FDA requests. So what we do is we have an injection into 30 animals with the drug, 30 animals with the vehicle, completely randomized and a masked trial. At 3 days, 1 month, and 3 months post-survival, the animals are sacrificed, and then we take blood for hematology and clinical chemistry, and we do gross and detailed necropsy. Of course, throughout the process, animals are weighed, there's clinical analysis and things like this, but for the most part, this is what a GLP study would look like that you would want a CRO to do, and this is what the FDA requires to take it to the clinic. So at the end of this thing, we have our own histopathologist, we have our own in-house histotechnologist who do all the processing, but we would get a report on all 60 animals that would report all of the findings of the study, and that goes into one of the modules in the IND application. So once that's all done, then the goal here is obviously not to treat rats, the goal is to treat the humans. So go back into patients who have early stage disease and inject this beneath the retina into the subretinal space, creating a bleb. Here's an example of what that would look like. Here's a colleague of mine, Steve Russell, injecting an AAV, in this case, Luxturna for RPE65, into the subretinal space, creating this little blister. So at this point, we've taken this whole program through all of these stages, and we're now at the FDA step, and have gotten this application reviewed and on the precipice of starting this trial in 2024. Well, what about patients like this? So this family, again, unfortunately, has 2 affected children with CLN3 associated disease. So the girl on the left here is now 11 years old. So this is what it looks like when you don't treat this disease in just 4 years. You go from having, a fair number of photoreceptors and 2300 vision into no light perception in both eyes. So she's NLPOU. So gene therapy is not going to help this young woman. What she needs is a cell replacement approach. So for the rest of the talk, I'm really going to focus on how we're using autologous induced pluripotent stem cells or patient-derived induced pluripotent stem cells to develop an autologous photoreceptor cell replacement strategy. And as you can see here, unlike our gene therapy approaches, which are just simply an injection beneath the retina, this is actually transplanting a full-thickness engineered graft. So this is actually work I did as a postdoc in Mike Young's lab, and we, this was me, Mike, and Henry Clausen, who's at Irvine, and we were taking sheets of photoreceptors. This was harvested from a pig, the one I'm showing you on the top, and transplanting them into the subretinal space of degenerative animals. Unfortunately, they would sit there for weeks and survive, but they wouldn't make a single connection with the host. And the reason for that, we discovered, was that as these animals progress, or as disease progresses and the photoreceptors die, you get this gliotic barrier, this gliotic scar that's formed at the outer limits of the retina. Interestingly, transplantation of stem cells, these were retinal progenitor cells that were harvested from developing retinas at the time. These cells have no trouble getting across that barrier. Not to get into all the details, but suffice it to say that they produce matrix metalloproteinases that allow them to degrade that barrier and just get right in. So we knew stem cells were the way to go. At the time, there was really only 3 stem cell sources. There were embryonic stem cells, which would need to be differentiated, of course, into retinal progenitors or photoreceptors. There were retinal progenitors themselves, which would come, we were taking them from about P0 mice when we were doing the mouse work. In human, it was taken from about a 22-week-old developing fetus. And then you had post-mitotic photoreceptor precursor cells. These were taken later in development and now are fate restricted to being only photoreceptors. Well, all 3 cell types had a huge amount of promise. The big problem with all 3 cell types is they're not immunologically matched to the patient. So then iPS cells came along and we really shifted our focus from embryonic stem cells and retinal progenitor cells into patient-derived induced pluripotent stem cells. And so as I'm showing you here on the very left, this is just a image of an iPS cell colony. In the middle here in panel B, what I'm showing you is an example of retinal organoids just 45 days post-differentiation, where we're starting to get this polarization and lamination, which I'm showing you in panel C here on the far right, of an organoid that's stained with some of these early retinal progenitor cell markers. If we let these things develop in culture over a series of weeks, this is, I'm showing you on the far left here, 150 days post-initiation of differentiation, these organoids laminate, with the outermost layer being where all your photoreceptors are. Interestingly, if you let them go for another 100 days, they don't change structure a whole lot, but they continue to develop and mature. So by 260 days, you see fully developed cones. Obviously, the outer segments are somewhat nubby. They're not perfect. There's no interphotoreceptor matrix really to speak of. But we have this very thick outer nuclear layer separated from the internuclear layer by an outer plexiform, which is sort of serendipitous as we're trying to harvest these cells in the outer nuclear layer for transplantation. Because although I'm not going to show this, partial dissociation allows us to get a highly enriched population of just photoreceptor cells for transplantation. And so we've done this, and you can isolate photoreceptor cells, transplant them into the subretinal space of again, rats. These are retinal degenerative rats. You can get, after 4 weeks, about 30 days post-differentiation, you get really good cell survival. You can get, these animals are obviously immune suppressed because this is a xenograft. You get integration. These are expression of things like recoverin. You can find the injection sites and travel out in both directions. All of the blue cells here that you're seeing are the host cells. This is an RD1 animal that has no photoreceptors left at the time of transplantation. We use these specifically to prevent issues with things like antigen transfer and concerns of that nature. But you can easily find cells that stain with human nuclear antigen that sit in the subretinal space and send projections up and make connections, synaptic connections with the inner nuclear layer bipolar cells. So what's the problem? Well, the big issue, as I showed you just in the start of this talk with iPS cells, is that they're really good models of disease. So if you transplant these cells without genetic correction, they're going to remanifest the disease phenotype that you use to test your gene therapeutics in the first place. So over the last 3 to 5 years, we've really, been focused heavily on the use of CRISPR-mediated genome editing. Back in 2017, when we published our first genome editing paper, we were using a standard CRISPR approach, whereby we would use a small guide RNA to target a specific location in the genome as close to the mutation as possible, co-deliver a Cas9 nuclease to make a double-stranded break, and co-deliver with that wild-type DNA. When you do that, you can then clonally select iPS cell lines that have repaired that mutation. This is not a very efficient process. However, because we're doing this in vitro in a dish, and you can clonally select the cell lines that you want and then do whole genome screening to confirm, it's a very viable approach. We've done this for a variety of genes. I'm showing you a different gene here than CLN3, MAC, and the reason I like to show this one is because this is one of the toughest genes to correct, because this is a 350 base pair allo insertion in the middle of exon 9. So using this technology, you can very robustly correct cells and select lines that have homozygous correction of something as complex as a big allo repeat in the middle of an exome. So now the problem is, as I've shown you there, I say we get good survival. However, that was not really long-term survival. Now, this is work out of Mike Young's lab, and one of the earliest studies that I was a part of after I joined this group. And what we showed back then is that a single cell injection or injection of a bolus of cells, photoreceptors in suspension, is really tough. You get a massive amount of cell survival. So you can get cells, as I showed you, but you lose upwards of 80% of them within days of injection. So what Mike showed, and this was his postdoc at the time, Minoru Tomita, at just 1 week, you get about 90% cell death following a bolus injection. If you were to inject the same number of cells on a scaffold, not just in suspension, you could bring that survival up to about 90%. So like a 9-fold increase. And that was sustained over this 4-week period they were looking at. So our ultimate goal then is not to transplant cells as a single cell injections, to transplant a whole graft, as I showed you with that cartoon when I started this section of the talk. The problem we had at the time is that all of the technology available to us did not allow us to make scaffolds that would promote cellular organization to anything that would look like a normal laminated retina. So we were using electrospinning, salt bleaching, templating, whatever was available we were using at the time. We could get nice sheets, but nothing that would ever look like this. So over the last probably 5-6 years, we've really focused heavily on this newer technology called 2-photon lithography. And what this is essentially a fancy 2-photon microscope co-opted to be able to polymerize a photopolymerizable gel at sort of the nanometer scale. So anything you can draw in CAD, you can print with this device. Now, this is our newest device. We just received this device a few weeks before the holidays, and now it's up and running. Because one of the problems with our first device is, I always use this analogy. You can build a house out of 1-inch lumber. It would take a long time to build that home. You really want to build it with 4x8 sheets, right? You'll do it in weeks. This is the same thing. If you are building a scaffold of 6 millimeters in diameter using 50 nanometer strings, it was really slow. This new version of the system, we can get our printing up about 80-fold. So now, on the order of printing previously, we could make 1 graph for a human would take us 7 days. This new system we can print in about 3 hours. So now we can actually do the studies we need to be able to take this to the clinic. So what are those studies? So the first things we need to do is actually, show that we can print with this device. We can just like our old device. print structures on sort of the nanometer scale. I'm just showing you strings here of 75 nanometers using this system, and we can generate any scaffold we want. This is one of the scaffold designs. These scaffold designs are constantly in flux because we're changing parameters such as scrollability, size, and shape. But regardless, we can now print large scaffolds to do the studies that we need to do clinic or preclinically in order to satisfy the safety requirements that the FDA asks for. So here's an example of 2mm scaffold. We actually print these scaffolds, take 1mm punches and cram those into the subretinal space of mice, which is somewhat of a challenge, but we have very good surgeons who do that. Ian Hahn is the surgeon who's leading the majority of that work. But you can see we can load these cells, these scaffolds, with photoreceptor cells. Here's an example of blue opsin staining, where we have this scaffold filled with blue opsin positive cone photoreceptor cells. You can see here, you can polarize these. We're putting them on sheets of RPE if we don't need RPE. If we're using RPE as well, then we plate the RPE first and put the photoreceptor cells on top. But as you can see here with the cone arrestin staining, these cells send axonal-like projections up. I say like because photoreceptors don't really have an axon, but they send outer segment-like projections down, which stain positive for cone arrestin. We've transplanted these into the subretinal space of pigs. As you can see here, this is actually a 5mm graft this is a 2mm retinotomy. So we built this tool that allows the scaffolds to scroll that we can inject through this small retinotomy. And we followed them over months. And you can see that these grafts actually sit in the subretinal space. They're very well tolerated. One of the reasons they're well tolerated is the material we use is PCL. It's very slowly degrading. So we don't get a very aggressive acidosis type of response that you get with some of the other polymers that are often used. And we've also gone on now to do this in rats. So the last study that the FDA really wants to see is truly the rat safety study, where we actually are now not doing a 3-month survival study for the stem cell work. They're requesting a 6-month study. So in this case, we put scaffolds filled with human photoreceptors into an immunosuppressed rat on an RD1 background, so it has no photoreceptors. And those animals survive for 1 month, 3 months, and 6 months post-transplantation. However, just like the AAV work, the FDA is not satisfied for us to make that product in the lab and take that into a safety trial. They want it made exactly like the way it would be made if it was to go into a human. So again, we brought this into the GMP space, and this was really the first phase of our GMP construction was this suite right here. This suite is a little different because unlike the AAV production suite where there's a single room with standard hoods, this suite is actually built around biospheric cell isolation systems. So the biospheric units are essentially atmospheric isolation units where we can control, in addition to CO2 and temperature and humidity, we can control oxygen tension. Because one of the things I hadn't talked about a lot up until now is we actually regulate oxygen tension throughout the differentiation, reprogramming, and differentiation process. So we drop oxygen tension during reprogramming, which increases reprogramming efficiency. We bring it back up when we start to get into retinal differentiation. As you can see here, this process takes 120 to 150 days. It's not for the weak of heart, especially when you're going into a GMP facility every day during the iPS cell generation and expansion phase, and then every other day during retinal differentiation. So the problem we are faced with now is not, can we make it? Is can we make enough to even do a trial? One technician probably couldn't make any more than about 12 of these grafts a year, maybe only 6. So our biggest focus over the next 5 to 6 years will be in this automation space. So what I'm showing you right now is an example of one of our robotic platforms that we've been developing called the Selects device. This was developed in collaboration with a group at Cleveland Clinic and Parker Hannifin, who's a big robotics company. We've housed these within side of biospheric units, as you can see here. This robot is really, a plate mover that's built over an automated microscope. You can see there's a stage. You can put plates on this stage. It'll image the cells for the entirety of the experiment. So you can put the plate on, image the entire plate, put it back in your incubator, follow the cells throughout the reprogramming and differentiation process. This is very useful because it makes it trackable, so we can track the exact colonies back to the start of reprogramming. The other cool thing with this robot is we built in liquid handling pumps, which are pretty simple peristaltic pumps, but this allows us to feed in an automated fashion. The other thing we built in is this syringe pump, which allows us to pick. One of the most challenging aspects of iPS cell generation, especially in a GMP space, where you're in a pair of coveralls, it's 37 degrees in the biosphere. It's very hard to do this when you have 3 pairs of gloves on. So having a robot that can come in and pick these iPS cell colonies, move them from the original plate into new plates, and then more importantly, be able downstream to pick organoids as needed for graft production is really handy. So that's one of the functions that we really set out to build into this device when we started. So using this device now, we've created on the order of 150 iPS cell lines. So sorry, more than that. So lines from 150 patients. We create 12 lines from every individual, and we keep 4 going, so 4 clones from each patient. So 4 times about 150 is what we've now made on this device. We've done, the standard validation. So here's just an example of morphology and karyotyping, as well as scorecard on the bottom. Cells are pluripotent. They're virtually identical to what you would make by hand, which is not really surprising because it's the exact same process, just run by a robot. If you differentiate cells made on this robot into retina, as you can see here, they differentiate as normal. We can get retinal organoids over a series of weeks. This is out to 120 days, this protocol. You can see at 120 days here, you're starting to get lamination of the retinal organoid with the outer nuclear layer staining positive for recoverant, and then the inner nuclear layer having early photoreceptor precursors remaining in there. So they are OTX2 positive cells. You can see here on the right, I just removed DAPI to show all of the photoreceptors in these organoids. If you let them go for another 40 days, so now we're out to, again, 160 days post-differentiation, and this is relatively selective. We're actually using organoids at this point because this is where we get sort of the most robust and largest number of photoreceptor precursor cells. You can see now not only are the organoids laminated, but the outer nuclear layer is laminated. So you have all of the rods staining positive for NRL here in the innermost limits of the outer nuclear layer, and all of the cones line up at the outermost limits of the outer nuclear layer. So again, with our sort of partial dissociation and microfluidic approach, which I'm not really showing you, but you can envision how you could enrich for cones or cones plus rods through this sort of erosion approach. And we fondly call this experiment the Tootsie Pop experiment, for anyone who knows what a Tootsie Pop was and how many licks it takes to get to the center of one. But that's what the strategy is. So if we then go take those cells, dissociate them, and do single cell RNA sequencing to compare cells generated on the robotic platform to that generated not only manually, but manually in another person's lab. So this was a data set published by Tom Ray and us. So we're in yellow, Tom is in purple, which fits with the universities. And you can see all of the same cell types were present in both cohorts in very similar proportions. So at this point, our last stage, this is our newest robotic endeavor, is incorporating a 6-axis robotic arm and a large automated incubator so that we can actually remove the human arm completely. So that all of these cells and plates would be stored in this big 500 plate incubator. The robotic arm would take them out of this little diving board that pokes in here. There's as you saw the flashing, a little barcode reader, so you can track each plate and each individual that would be in the plate. The robot will put that on the Selects device. The Selects device would then feed an image and then place them back in the incubator. So you can do differentiation over hundreds of days with up to 500 plates of cells by a single technician. So this is obviously not yet ready for clinical manufacturing, and we have a lot of work to do on this side. However, that's sort of the direction we see in the autologous photoreceptor cell replacement space. So with that, I'm going to stop and say thank you to a bunch of people, mostly the people in my lab who are involved in this. Kristen has reprogrammed more iPS cells in my group than anyone, probably has reprogrammed more iPS cells than anyone on the face of the planet. She's been with me since 2010. And has made on the order of 1,000 iPS cell lines or iPS cell lines from 1,000 different patients at this point. Laura is a very talented research scientist who did most of the robotic 3D differentiation work. Aaron did all of the CRISPR genome editing work. Jess Cook is a very talented developmental biologist in my group who's developing many of the robotic 3D differentiation protocols. And then, of course, a lot of our technical staff who run the GMP facility. And then really importantly, none of this work would be possible without the two gents that are with me in this photo here, Ed Stone and Rob Mullins, who, every grant we have written are all together. Everything we do is really this trio. And I couldn't have done this without any of this stuff, really, without these two guys. And then, of course, our surgical colleagues, Steve Russell, John Russell, Ian Hahn, who did all of the rat work. Tim Boyce is doing a bunch of our pig work with Steve and John. And then my collaborators in Cleveland, George Muchler, and our close collaborators and colleagues at Ohio State, Kim and Brad, who do all of our image analysis on the robotics. And then finally, our funding sources, R01s, and philanthropic support. So with that, I'm going to stop and hope I'm right on time and take any questions that anyone may have.
Several questions, and let me start first. I think again it was a fantastic presentation, and we're extremely grateful that you decided to join us today on that Friday celebration. My question is to your first part, the Batten disease. If I recollect, those patients have a very low life expectancy, right? And which organ is the most affected for
it's the brain. So the retina is, it's a very interesting disease because Batten disease is sort of a broad term. So CLN3 associated Batten disease is the version of Batten disease that ophthalmologists see. So the blanket term is NCL, or neural ceroid lipofuscinosis. So in that Batten disease is the one that ophthalmologists see. They first present with visual issues. So there's nothing else. They're perfectly normal, and they present with very rapidly progressive loss of visual acuity. So their photoreceptors are dying. That's the very first thing they see. However, they progress to having pan-neural degeneration. So they get seizures, very low life expectancy, cognitive decline. This all happens sort of later, though, by quite a long period of time. So from the time they're about 5 to 6 years of age, they start losing their vision. A lot of patients don't even start to have seizures until they're in their 20s. We have had patients who actually are very functional until they're well into their 20s. Unfortunately, many of those people will catch up, and there's rapid decline. But I would say the most sort of aggressive part of the disease is this pan-neural degeneration. Why it starts with photoreceptors, we have really no idea. But it doesn't only affect the photoreceptors, even in the retina. So in the retina, ganglia cells are affected, bipolar cells are affected. It affects, and even RPE, it's amazing. So Ruchira Singh from Rochester published an outstanding paper a few years ago where she's showing there's an RPE phenotype. And actually, we now use that RPE phenotype that Ruchira first published to test our AAV vectors, because it's much easier to make RPE than it is these retinal organoids. It's also much easier to transduce RPE in the dish. So it's not just neurons as well. So it's any cell that seems to be of neural lineage. Now, surprisingly, even though CLN3 is expressed in like almost every cell we've looked at, there's not non-neural phenotypes, or not ones we've really seen outside of the nervous system that are really a concern for this disease. Now, it's, who knows what would happen if they start living into their 60s and 70s. But at this point, brain rot is really everything central nervous system is really the issue.
Well, thank you so much. Let's move to our Zoom and maybe start with Daniel and then Brian.
Thank you. But beautiful talk and beautiful images, amazing facilities. So my question is about connectivity. A lot of groups try transplanting sheets of photoreceptors and maybe even whole retina. And we try too, and cells do survive, indeed, as you mentioned, but don't connect well. And I wonder, what is the extent of connectivity you see between transplanted photoreceptors and the host bipolar cells?
Yeah, that's a great question. It's low. I think we see the exact things you're seeing, especially with sheets of photoreceptors. We see no connectivity when you do mature sheets of photoreceptors. We do see, I don't know if I could give a percentage, because the problem is when we do single cell injection, our survival is also very low, right? So what the thing you're asking now, Daniel, is really what I think the next frontier in the cell replacement strategy. It's synaptogenesis. How do we break down the glial scar, get more of these processes from the photoreceptors to cross into the host retina and make synaptic connections with the bipolar cells? Because we're not looking, the problem with the field up until now is people have really focused on cellular integration, meaning they're putting cells in and then they're expecting those cells to migrate into an existing outer nuclear layer, right? And then make synaptic connections. Well, the reality is, if you have an existing outer nuclear layer, I'm not putting photoreceptors in, I'm looking for gene therapy, right? My goal is to keep those cells alive. So we're always, at least from our group, we're always been focused on the situation where there are no photoreceptors left and you're putting in new photoreceptors. Well, that's really challenging, right? So now we're talking about not the cell migrating from your transplant into the retina. You're talking about an axon being extended from your transplant into the retina. And as we know, that that internuclear layer and that barrier that's formed there is filled with chondroitin sulfate proteoglycans, right? Which axons really don't like to cross. So you got to break that down somehow. You also then got to stimulate synaptic connectivity with a bipolar cell that probably hasn't been talked to in a little while, right? So there's going to be a plasticity issue that we need to consider. So right now, I would say if we consider synaptic connectivity from the transplants we get, I say we're less than 10% of the surviving cells that actually make what I would call a synaptic connection, and we could detect a real B-wave ERG response. So we're going to obviously have to get that way up, but I'm not sure what the percentage of axons who are extending out of the graft is at this point either. So there's a lot of work that really needs to be done in that sort of classic neuroscience space. The one advantage we have over the ganglia cell people is that we don't have to ask these axons to grow, 6 inches back to the midbrain. So, and there's no myelin, which is great. Even though there's chondroitin sulfate, there's no myelin. So myelin being the most inhibitory molecule on the face of the planet, getting through that in the optic nerve is going to be a real challenge. So at least we're hedging our bets in sort of the easier space, if there is such a thing. But anyway, that was sort of a long-winded answer to a very short question of, we don't really know at this point.
Okay, thank you.
Hey, Brian.
Yeah, so Daniel, a bit of my question there, but I'll refine it as to we've done a bunch of work with some cortical organoids folks, and I'm wondering what the status of the circuitry is within your organoids, right? Do you have synapses? Are there excitatory neurons? Are there inhibitory neurons? Are there any gap junctions?
So that's a great question. It's so I can't answer the question because I have no idea. We know there's synapses. So if you stain them, you get an outer plexiform that's stained with things like, synaptophysin, VAMP, PSD95, all those things the proteins are there. However, we just literally 2 weeks ago got a rig from 3Brain. I don't know if you know 3Brain. It's this MEA-based system, but it uses sort of a CMOS sensor instead of a real microelectrode array, because it allows us to get 1,000 of these things. So right now we're chopping up organoids from a patient who has enhanced S-cone syndrome. So they have an exorbitant number of blue cones, and putting them ganglia side down, so just chunks of them, and then shining light on them to see if we can record activity from the ganglia cells by shining on different colors of light. Because in that case, what you should see is if you hit them with blue light, you should have a much sort of exaggerated response than if you hit them with a different color because they have enhanced S-cone syndrome. But that is an experiment that's absolutely critical to answering that question that I can't answer because we've not yet done it. I tell you what I have done. I've destroyed a couple of 3Brain chips that were sent to us within the first hour of opening them. So we got a lot of learning and sort of learning curve to do there. However, in the literature, there's a couple of people who have done similar things. So the reason we bought the 3Brain system is because Linda Lako from Newcastle had been using the 3Brain system. And what Linda had done is showed that if she dark adapts and then chops the organoid in half, dark adapt, and then put them ganglia cell side down on the 3-brain chip, she can actually record like spontaneous activity, but then she can record light-induced activity when she shines it on the photoreceptor cell side. So that at least makes me believe that they are connecting based on the fact that we see synaptic markers in there. And Linda has reported in the literature, recording from ganglia cells on an MEA. Jason Meyer has done a very similar thing with a different rig, and they see very similar results. So they can record from these organoids, sort of ganglia cell side of the organoid, by shining a light on the photoreceptor cell side. Now, gap junctions, I don't think anyone has looked anywhere nearly that closely at any of these organoids, sort of from an electrophysiology standpoint. I'm a very crude electrophysiologist, so again, this is sort of a shine light on it, see if you can detect activity. But I'd be love to discuss that more with you guys, because you know what you're doing in that space. I don't.
Right.
Send us some fixed organoids. We'll take a look at it for you.
No, exactly. So I think that would be super easy. We could do that tomorrow.
Cool. Awesome. Let's
Well,
talk.
if we get dug out, we're going to have 2.5 feet of snow by the time this talk is over, by the looks of it. So
Yeah.
I may not be able to send anything out of here, including me.
Right. Okay. We'll talk soon.
Let's go to Varuna. No, yeah, all right, we think that he has no good connection. So let's go to Vladimir then.
Yeah, hey, but very interesting, very exciting work. The topic of connectivity was discussed already. I was going to ask about that, but what about outer segments? You kind of alluded to the fact that in the scaffolds, you see outer segments, kind of black structures. Do you see some, and do you also see differences between rod and comb cells in terms of how efficient they are in growing outer segments?
We did. That's a great question. We definitely have looked at that, and we have seen that. So we get outer segments. So we get very nice inner segments in all of these organoids. Outer segments, I hesitate to call them, outer. I always say outer segment like. They do have discs in there, but they're not well organized at all. They're sort of, fluffy. There are people. So a couple of groups have really focused a lot on this in the engineering side. So Molly Shoykett, I'm not sure if you're familiar with Molly's work. Molly's a polymer chemist at a University of Toronto, and they've been focused a lot on developing a hyaluronic acid-based gel so they can promote outer segment development. And when they do that, it appears that they get much sort of more elaborate outer segment-like structures from these organoids. However, what we've seen, so again, we have a lot of experience with cutting through these and staining, but looking at the outer segment like that with the EM is somewhat challenging, because it's very easy to break all of these structures off, especially on the rod side. So I could be biased and say our cone outer segments look much better, but I don't know if that's because our cone outer segments are sort of stubbier and they're protected by all the rod outer segments. You know what I mean? And then we're getting artifactual processing is lopping off all of the rods. So all we're really seeing is nubby little pieces of the rod outer segment with a much more developed cone, which is what I'm predicting. So I predict, because if you look under phase, you get this really beautiful brush border, it's been termed. Now I'm trying to, at this point, very efficiently, which I know this is not going to work, look at to show you. So the most beautiful images, I see Rob on here. Rob might actually have access to it more than me. We've done scanning EM on these organoids, and using scanning EM, where you're not doing a lot of the processing, you can actually see very beautiful rod outer segment-like processes projecting up over the entire surface of the organoid. And if I can find that in a reasonable amount of time, I'd love to show it, because it's so freaking cool.
Very cool. Thank you.
All right, we have a question from David. David?
Hello, I just wanted to ask a quick question regarding the scaffold and the mechanical stiffness of the PCL structure, whether that could be an issue upon transplantation. And second question regarding the polarization of the photoreceptors that you mentioned, whether that is triggered by the RPE cultures or solely or potentially by the structure of the scaffold. Thank you.
Yeah, so those are two great questions that are right in my wheelhouse. So the PCL work, so this is actually why I went to work with Mike in Boston way back in 2006, because he was really pioneering the use of these sort of tissue engineering approaches with Bob Langer to develop scaffolds for cell delivery. And so we, over the years, have used every polymer that's not nailed down, ranging from PGS, polyglycerol cephacate, PLGA, polylactic coglycolic acid, PLLA. Literally every version of polylactic acid you could get, and PCL. So PCL is a very interesting polymer that we've sort of stuck with over the last 8 to 10 years for two reasons. It's extraordinarily well tolerated. So we put PCL scaffolds of 100 microns thick into the subretinal space of rats now for over 16 months with no sort of destruction of the inner retina. It sits on it beautifully. It's very slowly degrading. So PCL degrades through surface erosion. Problem with all of the PLL, PLGA approaches, they degrade through bulk degradation. So if you have a lot of the material, it degrades really quickly and it releases glycolic acid, right? So it's polylactic coglycolic acid. So you get a ton of acidic byproducts dumped into the subretinal space, and they're not well tolerated at all. If used in small amounts, PLGA is actually quite good, right? So Kapil Bharti's group has been using a PLGA backing for RPE transplantation, but it's a very thin layer and it has a layer of RPE on top, right? So here, having that very slow degradation and surface erosion has really prevented that sort of acidic byproduct destruction. The other problem with a lot of these polymer materials, so PGS being biorubber, we've used a lot of that, also gives a, so you use essentially glycerol and sebacic acid to make PGS. So it's very easy to make. You can make it in your lab. Takes like a day in an oven that goes up to 120 degrees. It's really simple. It's a really attractive polymer because it's called biorubber for a reason. You can roll it up, let it go, and it unrolls perfectly, right? It's super stretchy. Problem is it also degrades quickly, releases sebacic acid, and for some reason that we don't know, that thing we call the cheese grater. If you put that in the subretinal space, and we've done this in pig, it ends up in the vitreous every time you put it in there. It just wiggles its way. It just destroys the entire retina. So of all of the polymers we've used, PCL, which is the slowest degrading, has been the best. Now for polarization and stiffness, PCL, we changed the stiffness, based on two things. So there's two stiffness concerns, right? So from a biology standpoint, I would like to have the stiffness of the material be as close to that of the native tissue, right? Problem with that is that the native tissue is retina and it's sort of like snot, the handle. So giving that to a surgeon makes it extraordinarily hard to transplant. So there's always this pull between what the cells like and what the surgeons like, because I could make it so that the cells love it, but then the surgeons just can't put it in. So there's always been this balance of how stiff can it be and still be handleable. So this thing, interestingly, we've done a lot of testing on this over the years, and we've tested the compressive modulus of the retina from pig and human. And although this material is still high, it's not orders of magnitude higher. So we're on the level of sort of 100, pascals to a kilopascal versus megapascals, which PCL, PLGA, and those things are. So the retina in compression is on the order of 50 pascals. So it's a very soft tissue. So if you get up to that sort of 1,000 pascals, and you can do that simply by using different molecular weights and polymerization parameters for the PCL, right? So we can cross-link this more or less to make it stiffer or softer, and we can use longer chains or shorter chains to make it stiffer or softer. So that's very controllable, and we've now ended on a material that's sort of, soft, scrollable, and very well tolerated in the subretinal space, and actually photoreceptors love it. So that image I showed you there was polarized. That was not on a sheet of RPE. They will actually polarize like that on their own from being in these sort of wells in the scaffold. Now, if you put them on RPE, then, that's the ultimate goal to really get the polarization and expression, because if they're completely devoid of RPE, they don't tend to differentiate as well.
All right, let's move to Emma.
Hi, Dr. Tucker. Wonderful talk. I work on the Better retinopathy, so of course everything related to retinal vasculature catches my attention. You mentioned it at the beginning of the talk that the photoreceptor degeneration will cause thinning of the vessel. When you do your transplantation, have you looked at the retinal vasculature? What happens to it? Does it come back or?
Yeah, so the interretinal vasculature is actually still there. It's just it's constricted, so it's auto-regulated. So if you put stuff in the subretinal space to suck up the oxygen, that retinal vasculature comes back. It opens up again. Now, the bigger problem we have on the vascular side, which is where I thought you were going with the question, is that in an AMD, Rob and I have, a couple of our R01 grants are actually focused on this very problem. In AMD, we have the flip side. So instead of the photoreceptors going first and then losing RPE and choroid over time as death and destruction ensues, there you have an early loss of the choroidal vasculature underneath the retina. So a very interesting disease, choroideremia, really shows this function. Okay, so choroideremia patients are very interesting biologically in that if you look in there, they have no photoreceptors left. Their interretinal vasculature is not constricted. Why is that? Because the RPE, okay, where the genes express, causes loss of the choroid, the producers of all of the oxygen, before the photoreceptors go. And once you lose choroid, you lose RPE, then photoreceptors die. You don't get auto-regulation of the interretinal vasculature because there's no oxygen coming across there. Right, so that's where biology really shows us this very cool biological phenomenon. So the interretinal vasculature doesn't go away, it's just really constricted because there's too much oxygen and it's just sort of shriveled up. If you block all of that oxygen, it comes right back.
John.
Hello, thank you so much for this talk and amazing research and facilities. I was wondering, with the attempts to do things in-house to reduce costs and overall reduce the burden financially, how close do you feel you are to getting some of these treatments to people?
So that's the $1 million question that we get asked a lot. I can tell you on the gene therapy side, I'm very optimistic. We're going to have 1 to 2 of these going in 2024. So we're starting with MAC. There's very specific reasons why we started with MAC, and that's followed up by CLN3. We actually had started first with CLN3, and halfway through we changed some of the vector stuff because of transduction efficiency in the interretinal neurons. So I suspect that we will be treating patients on the gene therapy MAC side within 2024. On the cell therapy side, we've been back and forth with the FDA multiple times. I would hope that we could get the first phase going in 24, but that's a much more complex program, and predicting anything with the FDA is like trying to predict the weather in Iowa, right? So it's a bad idea. You should just like working on a grant or a paper, when you get comments back that that were a little salty, you should take it and do everything you can to address those comments and put it back in. So it's a very sort of iterative process with the FDA. The thing I will say on the FDA is there's also a steep learning curve there. And thankfully, we've gotten over that. So we now know the language, we know how to deal with the FDA. And I'm very optimistic that in 2024, vectors and therapies, drugs that have been made in our facility will be put into patients in clinical trials for these rare inherited eye diseases. So that's sort of where the state of play is at the moment. But on the cell side, it's very hard to predict any of this stuff. But it's not, we're not talking 10 to 15 years anymore, right? Now we're talking sort of months to single-digit years even on the cell therapy side for early trials.
All right, Priya.
It was an impressive talk, Dr. Tucker. So my question is more related to Dr. Kefler's question. So I wanted to ask you whether you noticed a lag in the development of rods compared to cones. And I'm asking this because rods, they require well-developed, organized RP layer. And did you observe a well-developed, organized RP layer throughout your organoids or no?
Yeah, so that's a fantastic question. So I don't know anyone, people have tried to get a well-organized RPE layer on the outside of an organoid. To my knowledge, that's not yet been achieved. People have put organoids on sheets of RPE. We've done that. That helps. But to really answer your question, it's interesting. We do see rod photoreceptor precursor cells come in quite early, right? the firstborn cell we see in the outer nuclear layer is a cone. So we see a blue cone first. So it does develop very similar to what a normal, vertebrate would develop. Then we get rods, then we get red-green cones. However, there does seem to be somewhat of a delay in the rod space. So the data we have on this, again, to use NR2E3, a lot of these answers are based on other diseases, right? So we have a program, a really cool modeling program focused on NR2E3, enhanced S-cone syndrome. And what you see in enhanced S-cone syndrome, if you block NR2E3, so these patients are homozygous for this mutation in NR2E3 really early in the gene. So if you block or lose protein expression for NR2E3, what happens is that it's not what we expected. What we thought based on the mouse is that all of the cells that were going to be rods, because this is a nuclear hormone receptor absolutely required for rod genesis, we just thought it'd be like NRL and they were all going to become blue cones. They don't actually. You get a whole population of cells that we've called them divergent rods. So they actually express some rod genes and some cone genes, but they're much more rod-like. They just don't express rhodopsin. They do express things like PE6H and GNAT1, which are, cone and rod phototransduction genes. However, the timing is very similar. So it doesn't seem like the time course of photoreceptor cell genesis is shifted by the absence of RPE. It seems like the maturation is shifted. Now, that said, if you take RPE out completely, our organoids suck. So there's not like there's no RPE in these cultures, right? Every culture, and what we've done in cultures that are notoriously bad is we've added factors like PEDF, and it actually does accelerate photoreceptor cell genesis and number, right? So RPE, I believe, are absolutely critical in this process. They're just not in this sort of monolayer on the outside of the organoid that we wish we could make, right? I'm not saying that won't ever get to that point. People are trying it where they're doping in RPE, sort of the coat, put a candy coating on this little Tootsie Pop on the outside. And that's been challenging to date. I don't know that anyone has really accomplished that in any meaningful way.
All right, we have one more question from the Zoom. Ben?
Okay, yeah, I wanted to get back to the question of making retinas in humans and getting FDA approval. What do you think is going to be a good outcome measure that you can practically use in somebody with very low vision? How do you demonstrate functional vision in these?
That's a fantastic question that we've talked about more than I care to admit. So we have, this has really been very challenging in the cell replacement field, right? So what I'll call, metrics or whatever the FDA jargon of the day is, really what the FDA is concerned with is this really a function of the new cell, or is this a function of the new cell preventing progression of the disease, right? So is it truly a cell wiring up and restoring function, or is it a cell going in and acting like a gene therapy type of thing and preventing progression? So what we reasoned back very early on is that, look, we're going to do first trials in people who are no light perception, okay? So then our metric is not going to be, are you aligned better on an EDTRS chart, right? Because that is fraught with problems. If you inject cells into a person who's 2100 and try to prove that that person has better vision, that is going to be a challenge. And that's where the whole field has been sort of swindled by charlatans who claim efficacy when there is none, right? Because it's very easy to see a patient in a clinic whose ERG is 20% better from one visit to the next, who is 3 lines better on an EDTRS chart from one visit to the next. Chris Johnson will tell you, the amount of variability between 2 visits in the same patient without any treatment is huge. Now, if you're no light perception, you have no photoreceptors left, and we put this in and you get light perception back, then I think that's something you would start to believe. If now you get edge detection back, that's something I would believe. But that's where we're starting. None of our patients are going to be anyone who have anything better than no light perception in the early trials.
Thanks.
For those very reasons.
All right. Again, Bob, thank you very much for your presentation and being with us today. The bad news is that Bobito showing his face rather than his dog, and that is very sad. And the last question goes to Elliot. And again, Elliot.
Oh yeah, thanks for a wonderful talk. What's the ratio of concept types in your retinal organoid, and do you get a consistent ratio from each lines?
That's a $1 million question. We don't control it actually. So we eventually will, we would like to, but at this point we take all comers. So we're taking all photoreceptor precursor cells, which is a mixture of rods and cones, and that's what we're filling our graph with. So interestingly, some patient organoids are much better at making cones than others. And so we don't quite understand that at this point. That could be genotype, background effect, whatever it is. However, that means that then your graph, because it's per patient, is going to be different than the graph from another patient with relation to the percentage of rods and cones. However, what we can say is we're upwards of that sort of 95% photoreceptor precursors. So our goal is to only put in rods and cones, or largely only put in rods and cones. However, we're not in any way at this point controlling the percentage of one versus the other.
Thank you very much.
Thank you, everyone, for coming.
Next Friday, 8:30, the same place, the same time. This is good.
Well, Chris, before I let you go, you see Rob actually was holding up your grant.